everybody. So glad to be here in New Zealand. And the weather is great anyway. All right, so I will talk about the cohorts that we have in Los Angeles. And so I'll just go through them. I don't have to talk too much about this because this is what Aubrey just presented for the Da Vinci study, which is the equivalent of SHIVERS-I. And so we have a very similar study going on in the US where we're recruiting the infants. We try to get them at birth, but we definitely get them when they're under 6 months old. And then we follow them over time, try to catch any illness visits. We also see them for annual visits so that we can, just in case there was an illness in there that we weren't able to see. And then we also have about 50% of our children get vaccinated, so we also see them pre and post vaccination. So we began this study just before COVID and did have a little bit of trouble enrolling at first. About 270 kids enrolled at this time. We did have some withdrawals, mostly during that COVID time where we had to shut down everything. But the illness visits, so since we didn't have a lot of flu at the time, we only have 4 flu A's, 1 flu B, 52 with SARS-CoV-2, and 10 with RSV. Those 10 were all this season. And then we had our household exposure and transmission study. This is the equivalent of SHIVERS-IV. And so we called it Hearts just so that we could make that logo for our families. But in this study, we enrolled the entire family and followed them during the acute period, tested them multiple times, all the family members, to be able to catch anybody that that would get infected. And then at first it was really just a transmission study. We wanted to know who got infected within that family during the immediate time period. And then as the study went on, we added visits every 3 months so that they would keep coming back and we'd be able to see them again and again. And so we kept doing that until we ran out of funding, which was in August 2022. But there's some good news that'll come out of that I'll let you know. But overall, we had 208 households that participated with 907 people in those households. The households range from 2 people in the household to 11 people in the household. And so it's about half adults, half children. And we captured 1,314 illness episodes. And even with the very first episode, we had 61% with confirmed SARS-CoV-2 by PCR. So very high numbers in this cohort. So with our household cohort, we captured quite a few illness episodes of SARS-CoV-2. And we were able to see not just first episodes, but second episodes, third episodes, and even fourth episodes within a household. And so I'm going to show you our next slide looks a little bit busy, but let me walk you through this because I think this is actually really interesting data. So thanks to Richard Webby, he sequenced the viruses that we isolated from these illness episodes within the families. And so we could tell with each the circulation of each of the viruses, each of the variants. So first we have, in the different colors, you could see I combined wild type epsilon and alpha just because we couldn't exactly tell where there was so much overlap during that time period. And then delta in green, Omicron in pink, and then the Omicron variants in blue. And so, and then the second column gives you the episode number within the family. So in the first episode, during the wild type period, we saw an attack rates of around 72% within our families. Our families that we enroll are relatively low income, live in we had multiple 11-member households that lived in 1 or 2 bedroom apartments, just to kind of give you an idea. And then if you go across, you can also see household vaccination rate at the time. So during the wild type, we vaccines weren't quite out yet, so vaccination rates were very low. It was just starting. And then if they had history of past COVID infection, and that's by our own study. So with second episodes during the wild type epsilon and alpha period, you could see attack rate was much lower, 29%, but that's because 42% of the within the family had already had previous past infection. And again, this is all detected by us, so all PCR confirmed. With Delta, you could see that the attack rates went down just a touch, not that much. Vaccination rates went up, but these were also still first-time infections. But with second episodes, that attack rate went down. And then you could see by the time we got to Omicron, the vaccination rates were higher, except in those families where you saw the second and third infections. And that's within the family, and that's because they weren't getting the vaccine, and so they were going through their third episode in the family. And with the variants, we started to see fourth episodes within the family. Now, for all of these illness episodes, we've collected blood at each episode, all the swabs, and all of the and then all the metadata. So we have information for each time these families got infected. And so with that, there's a lot of data and a lot of samples to work with. So I think that that's pretty exciting. So this is another cohort that's kind of separate, but at the same time, this is a vaccination cohort for SARS-CoV-2. So this cohort began in December of 2020 when the vaccine came out. And so there's a lot of overlap with the heart study in terms of how we set up this study. So we, for this cohort, we obtained blood samples right at the baseline before vaccine, and then followed them from there every 3 months. And so we are curious to see what happens to them every 3 months after each vaccine, after each booster, after infection. They all come in for if they're ill. And so we want to know how their immunity changes with each time that they're vaccinated or gets infected. And the really neat thing about this is that we also have a blood sample before they get infected. So we can actually see, and many of these have the whole household enrolled as well, so we can see their blood, what kind of immunity they had in their blood before infection happened. And within that household, for those that didn't get infected, we have their blood as well, so we can see what protected them. And so some of the hearts cohorts, some of the hearts families that have been consistent in their follow-up, we will roll over into this study. And this study is just newly funded by NIH. So far in this study, we have 482 people enrolled of all ages, as you can see. And so right now we're enrolling down to 6 months. So it's everybody where we began enrollment for each age group when the vaccine was available for that age group. And then because we're talking, and then I wanted to mention that 32% of this cohort had history of prior SARS-CoV-2 infection, and we know that based on pre-vaccination serology. So what that means is, close to 70% had no history of SARS-CoV-2 at the time of vaccination, and so it's an interesting, will be an interesting cohort to follow. And then since we're talking about cohorts, I threw in one other cohort that we also lead that's separate. This cohort follows for diarrheal diseases, still viruses, and it's the goal of this cohort. So I'm a pediatrician, and my interest is what protects these children clinically from disease. And so rotavirus vaccine works really well in some countries and not as well in others. It usually works well in high-income countries and not as well in low-income countries. And so we're curious what's the difference between the two countries in these children. And so we enroll these children at birth. We see them during the newborn time period, and then we collect, and then we see them 2, 4, 6 months, and 12 months. We collect samples, including blood, milk from mom, stool for microbiome studies during these time periods. And so, again, here we combine clinical data with the data that we can get from the specimens. That includes the microbiome data, antibody data, T-cell data, and we're trying to figure out what leads to those high responders versus low responders. So, and there's 520 mother-infant pairs enrolled into this cohort. So I just thought, there's just so many areas where we can collaborate, even if this cohort isn't looking for respiratory infections, but maybe we could add that, for example. Anyway, with that, I just wanted to thank my team and the many, many collaborators around the world that make these cohorts possible and make them happen. So, and then I forgot to put NIH in there, but thanks to NIH for the funding. So, thank you.